welcome to the second day of this wonderful symposium. Thanks again to Chirag and Professor Devnath for putting this together. Very nice to have you all back here. So without further ado, I'm just going to call my colleague Manikandan. So keeping with the tradition of not giving long introductions, I'm just going to call Mani onto the stage. But you can always look up his CV online. So Mani.
Thanks, Karthik, for keeping it brief. Yeah, so I can get on with the work. So it's a pleasure to be here, and thanks for the invitation to Devnath, to Chirag, and other organizers here. It's been a wonderful event so far, and I hope I can add my own bit to the event that happened yesterday and that will happen later today. So I'll talk about. Are you able to hear me in the back here? Okay. I'll talk about two excursions into inter-organ computational biology. That's what I call the kind of one arm of the work we do in our research group. And you can see members of our research group, old and new. Some of them are continuing. And so I'll be discussing about work by two of the members in my group and in collaboration with others, but two lead members. And this will be about predicting molecular mediators of inter-organ communication. Okay. So these are molecules such as hormones and the genes which are involved in the processing and release of the hormone or in the response to the hormones. Okay. So that is what we will look at, and then I want to take two different routes to do that. One is analysis of literature data. Biomedical literature, we know in PubMed we have 20 plus million abstracts and articles, so how do we mine that data, right? And then the other one is omics data, so genomics data. By genomics, I refer to not just the genetics. So yesterday, genomics was mostly used to refer to the genetic material, the DNA. But here we'll be mostly looking at gene expression data, the transcriptomic data or the genomic data. Okay. So those are the two routes that I wanted to talk about. And as I said before, this is just not for the namesake I'm saying. So because I have a connection to this particular building, I started my research career as a summer intern. Right across the hall in Professor R. Govindarajan's lab. So it's a good reminder for me to be here. I have not spoken here before. We have had like group meetings in Professor R. Govindarajan's lab, and so I've had this several years ago before my hair turned gray. And so basically it's very nice to come back here. And then the memories I remember from there as I was a junior researcher. So this talk is targeted more towards, yesterday when I was looking at the posters, most of you PhD students, so this talk is targeted more towards you. And so this is, as Professor Narahari also says, this is like a temple he refers to, IISc campus. So it's a temple of knowledge, and it's a great place to have you exposed to these kind of research discussions at this place. Okay. And these two excursions are joint works with Aditya Yadav, who is the first author of this work, along with Tarun and Mohit and Tamirini. The second work is joint work with again led by Tarun, and joint work with RS, we call him. So RS is from actually Intel, so some from Intel may recognize him. So he's from Intel in Intel here, and Sangamitra, who is an excellent postdoc in our group, and she kind of brings in a whole wealth of biological knowledge to whatever computational work and predictions that we make. And of course, this joint work with Ravi. Tarun is my colleague Ravi and my joint student. So this is kind of a holistic picture that I wanted to start out with. So the interactions are of the essence in biology, biomolecular interactions that I hope all of you, doesn't need kind of preaching. So you are in this area, and so you would know that genes do not act in isolation, proteins do not act in isolation. They all interact with each other. And a molecular level that is shown on the right is typically what most of computational biology, or things are changing now, but most of computational biology is focused on. You take a particular tissue or an organ, you do certain genomic analysis on it, you look at the DNA or the RNA and the protein content, the proteomics or transcriptomics, or the genome-wide DNA content, and then you do certain analysis with it, right? And but to understand what is happening within the whole body, we need to take a holistic viewpoint, look at all the different organs, how they are interacting with each other. But the challenge is, can we do it both at the holistic and also at the molecular level, right? So these two different these two form different scales, and so can we really reconcile both of these different scales? And what I claim is we can do so, yes, we can answer, we can obtain a holistic yet molecular level map by looking at these gene-gene interactions, by modeling it as a connection between the different genes in the different tissues, right? So here you have 3 different tissues. So it could be the brain, the heart, and the stomach here. And then you could have like these different tissues. The same set of genes are present in all, but they are at different levels and they have different connections. And some of these connections, we know the causal relations. So G3 is causal for G4. Some of the other connections are just simply correlations or associations. So given this incomplete understanding of the system at the gene-gene level, can we infer something holistic about the system, right? So what are the genes that are participating in intertissue communication? So here, for instance, G2 is participating in these orange edges. These are these intertissue or what we call interlayer connections, and the blue edges are the intralayer connections or intra-tissue connections, right? So here it seems like G2 participates in many interlayer connections, and G3 also, because it's controlling G2, it has an indirect effect on the genes in the G4 and G5 in the tissue B, right? So these are the kinds of questions we want to understand. Can we build this network in the first place, and can we analyze this network so that we can look at what are the key players that are involved in intertissue communication, okay? And this has been, this is not totally new. This is relatively new compared to within tissue genomic analysis, but it's not, of course, totally new. I mean, hormone biology has been studied for several decades, right? And we know a lot of different hormones and the genes that are involved in the production and response to those hormones. And recently also a genome-wide view is taken up in terms of experimental approaches, and there is more and more interest in understanding intertissue communication network. They call it the ICN by Norbert Perryman from Harvard and people who work in this particular area. And especially they focus on experimental efforts in model organisms. So they start out with Drosophila, which is easier to do genetics on, right? And then they are now followed up efforts on mice, where they try to figure out not just standard canonical proteins or canonical hormones. Are there other peptides or other hormones or other molecules involved in intertissue communication, right? So that play the role of hormones. And so those are some efforts, some example efforts that started out with Drosophila in 21, and then they have now moved on to mice in 2022 mostly, and where they are looking at labeling proteins in a particular organ, in an organ specific fashion, and then they see where all it goes, right? How it circulates in the blood and what are all the different organs it reaches. And then they look at what are the new proteins that are not known as hormones before, that are traveling from one region to the other region, right? So for example, from the brain to the muscle, or from the fat body to the different muscles, okay? And so there are many different metabolic diseases that can be studied using a intertissue communication network. So many diseases, we study it mostly focused on a tissue because of tractability, but many complex diseases affect multiple tissues. I mean, diabetes is one example. And so we would like to understand a holistic, a whole body model, okay? But our particular interest, I mean, I've never done wet lab before, so our particular interest is in computation. Can we use the power of computation to tease apart and analyze these interorgan communication networks, or ICN, right? And specifically, we are going to focus and narrow down on the problem. There are many open questions in mapping and analyzing ICN, but we are interested in predicting hormone gene relations that constitute this ICN, right? From, like I said, these two routes, data mining from literature data mining as well as genomic data mining, okay? And these are specifically multi-tissue genomic data, and what I mean by that is from the same individual, right? So for various, post-mortem samples of different tissues from the same individual collected across hundreds of individuals. So this is like the GTEx data that a speaker from SBA was talking about, right? So we have already heard about the GTEx data, NIH GTEx data, which has across hundreds of individuals, they have collected different, harvested different tissues and performed RNA-seq gene expression analysis on those individuals, okay? So we'll do it in this order. So in order to predict these hormone gene relations via literature mining route, some of the motivations, I've already told you that we have about 20 plus million abstracts, and then it keeps growing by millions. So a single person cannot read all of those articles and try to infer all of these hormone gene relations. it has a treasure trove of relations, both directly embedded in the literature, right? So these may be in the same sentence. So insulin gene is binding, or the insulin protein binds to the insulin receptor, right? So then you say that insulin is associated with the insulin receptor gene, right? So if you have a sentence where two terms, the hormone and the gene, simply co-occur in the sentence, that's a direct link between them. So those are those we can extract. But can we also extract those that are indirectly hidden? So insulin and maybe some other gene XYZ may not be directly referred to in the same sentence, but then insulin is often cited along, co-occurs with some other sets of genes, and then the other XYZ gene then has the same neighborhood, and the neighborhoods of these hormone and genes overlap with each other, right? So then that is something what is indirectly hidden in the literature. So can we mine the literature to predict both these direct and indirect relations? Okay, so that is what we are interested in. Our contributions in this area is we are essentially, there has been a lot of work before on predicting disease gene relations, disease drug relations, and so on. So we are the first to focus on predicting hormone gene relations, and this is possible because of two things that we did. So one is that it is enabled by expressly, so just for this purpose we constructed a hormone gene database, a ground truth set of hormone gene relations, right? Because of this ground truth, we then analyze the properties of this ground truth hormone gene relations, and we figured out that we need to do certain class, to address certain issues of class imbalance. We will get into it in a short while, and we propose a balanced embedding approach, a word embedding approach to predict the hormone gene relations. Okay. Like I said before, I wanted to target it towards more of the junior researchers, so mostly students here. So when we started out this project, there was no hormone gene relation database, or at least not in the format that we wanted. There were a couple of existing databases, but they were in different formats that could not be used to train our model. So we just basically rolled up our sleeves, and three of us, like Tarun, myself, and an alumni from our group, Arjun Sarathi, who is currently doing a research work in Europe. So we basically pulled our sleeves and said, okay, every week, all the days we'll just look at some of the gene ontology terms and then figure out which are all the hormone gene relations. So do not expect that all the data sets would come clean in an earlier paper. So be ready to prepare the data sets yourself. So not everything comes in a nice Kaggle contest, right? So research is very unstructured. In fact, there's a kind of informal code that we say that 80% of the work is in preparing the data and cleaning the data, and then 20% of the work you do the rest of the research. Okay, so that's something that I wanted to communicate to students who are just entering this area here. So we were able to build this HGV1 database. It looks like the following. So you have like certain hormones like glucagon and adiponectin. We had to go through different resources to figure out what are kind of the canonical source tissues. So glucagon is produced in the pancreas, it is received in the liver predominantly, and you have certain genes which are associated with glucagon secretion, so in gene ontology terms. So we scanned through all the gene ontology terms, and we figured out that these are certain terms, glucagon secretion and glucagon processing, that we could term as source genes, right? So all the genes annotated to those terms, we can call it as source genes for glucagon. And then the target genes are those that are response to glucagon receptor activity or binding and so on, right? So we essentially went through all of the gene ontology terms and then figured out which ones are hormone related and which ones are hormone source versus hormone target, right? And adiponectin is another example that is given below. Okay. So after we were done with that compilation, so when we looked at the number of genes associated with the hormone versus the number of hormones, so this is a simple distribution. You can see that there are some hormones which are associated with 500 genes. Can someone predict what this is? What hormone this is? A hormone associated with 500 genes, but many others are like less than 100 genes, some with 200 and so on. Insulin, yeah. So it's insulin. Insulin is a well-studied hormone, and so there were like some hormones that are very well-studied, insulin, or some medium studied like 200 genes. So I forget which hormones were around 200 genes were associated. So there is a class imbalance here, or not really class imbalance, but a hormone imbalance, right? So there are some well-studied hormones, some poorly studied hormones. So we need to balance this data set. Otherwise, whatever model we build will be just dominated by insulin genes, and so it will just predict genes associated with insulin as hormone gene predictions, right? So to do that, one challenge is that we cannot apply like there is SMORT approach that Karthik was referring to earlier in yesterday's talk.
So there are approaches like SMOTE, but we cannot apply it directly on text data. We need to convert it into a numerical vector representation, before which we can apply these standard machine learning methods, right? So what we did was, by the time we were working on, I mean, the machine learning field changes so rapidly. By the time we were working on, word to vec and other embeddings were quite popular. So we just converted words to numerical vector representations. And so here is an example that is shown here, where there are two harmons, right? So one is this triangles and another is this dot. So the dot is the more well studied harmon and the triangles are poorly studied. The two colors refer to genes that are associated with the harmon, and the black is genes that are not associated with the harmons. Okay, so there are the positive and negative class. You want to separate, you want to find the boundary between associations versus non-associations. These are, each dot here or a triangle here is a harmon gene pair that is related, and then the black dots are those that are not related, right? But we want to make sure that the number of triangles and the number of dots are the same. So then you can convert this into embeddings using standard representation learning approaches. So at that time, like I said, word to vec was a popular approach. These days we would have used BERT, right? And then by these days I mean in 1 year later on, right? And then in months these representations keep changing. So we would use these embeddings, and once you have converted into embedding, right? So the idea is that then you can balance it in the embeddings space using approaches such as SMOTE, okay? Which I have a brief slide that tells you the rough idea behind it. And so now you have balanced out the number of genes associated or non-associated with each of these two harmons, and then you use a standard support vector machine. Because our data set was not too huge, you can see here that the number of relations was not in the thousands or millions, right? So we will have to resort to some classic ML approaches. We could not, or it doesn't make sense to use a deep learning approach. A simple support vector machine classifier worked very nice in separating the true relations, true harmon gene relations from the non-associations, non-relations, okay? And this is where I said, in terms of the whole workflow, we have this data set which has these harmon gene relations, and then we take some random pairs as harmon gene non-relations, right? And then all of these are combined together, and we do proper cross-validation and other things, and we convert this harmon and gene symbol into these 200-dimensional embeddings called BioWordWork. Actually, we tried, like a reviewer asked us to try BioBird, and we tried that, and BioWordWork actually performed better than BioBird. So there are always utility. There's another tip for students is there's always utility in going back to older methods. Don't just take the latest greatest method. Also compare a few versions earlier when you compare these. So for us, actually, BioWordWork is how we use these derive these embeddings from the harmon or gene terms. And that gave these 200-dimensional embeddings, which we can concatenate and then balance it and use an SVM classifier that perform quite well with roughly an average accuracy of 70% for predictions. Okay. And you can actually visualize these word embeddings. So before we actually trained SVM model, which is a supervised machine learning approach, we wanted to try an unsupervised approach to see if there is signal in the data, right? And we could see that all of the harmons, if we cluster it according to the similarity between their embeddings, each embedding is essentially a vector representation of a harmon or a gene, right? And so you can look at these vector representations, you can compute the cosine similarity between those vectors, and then you can use those similarities to group those that have high cosine similarity together and low ones farther apart. And I guess wanted to just point out to you a few things. So maybe there are these follicle stimulating hormones, thyroid stimulating hormones, and there are these reproductive hormones that group together. So there is oxytocin, relaxin, these are muscle related hormones, so gonadotrophin. So all of the so there were examples of hormones that group together according to the known biology. Okay. And then if you look, kind of dwell deeper into how certain genes are associated, I talked about these indirect associations and direct associations, right? So you may find certain things that are co-occurring in the same sentence, so that's a no-brainer. So you just then attach a hormone to a gene, but there are others in which the neighborhoods may be overlapping even if the hormone and gene are not in the same sentence, right? So here you can look at the neighborhoods of these particular genes and the hormone estradiol, right? So the estrogen hormone, and you can see that there are certain genes which are common in the neighborhood. So the neighborhoods of these genes and the neighborhood of this hormone are overlapping. These are the red ones, and of course, I mean, there are these different genes that are all in the common neighborhood of these hormones and the genes. Okay, and similarly, you can also look at certain other factors, not just genes that are in the common neighborhoods, but certain other terms that are in the common neighborhoods of the same hormone and gene pair. So GSTP1, for instance, is often co-occurring with hormone refractory prostate cancer, let's say, and then that estradiol is also co-occurring with the same term. Okay, so there is a similarity in terms of where they lie in this embedding space, and these are all the common terms in the overlapping neighborhood of this hormone and gene. So that gives you the signal to predict hormone gene relations using just the embedding or the vector representations of these hormones and genes. So that leads you to wonder if these unsupervised approaches based on simple similarity between these two vectors, a vector for the hormone and vector for the gene, gives you reasonable results. Would that perform well, or would supervised learning perform better? So we do see that these dotted lines are unsupervised learning based on simply cosine similarity and some cutoff on that, right? So you just take the hormones embedding, the genes embedding, you look at a similarity between those two vectors and then see if it is greater than some value, right? And then for different values you see this ROC curve where you plot the false positive rate versus the true positive rate. The more closer towards this corner the better, and so you see that these thicker lines, which are the supervised approaches, which is our bioembeds approach based on this SVM classifier, does better than unsupervised approaches. Okay, and we have done certain cross species validations where we took some mouse data that was entirely out of the training data that we used, and so we took basically the model that was trained using human data, and then we applied it on the mouse data, which is the test data, and then we did find that it also performed well in the mouse data, and it was not driven by similarity between the positive mouse genes and the human genes. Across different levels of similarity you get varying levels of accuracy, but then they are not just driven by those gene symbols that are similar between mouse and human. So even for gene sets that are not similar between mouse and humans, our models still work better. Okay. By the way, please stop me if anything is unclear. Okay. Okay. So we can assemble our predictions and construct this intertissue network that I was talking about, right? These hormone gene relations are one main component of this interorgan communication network, and we can look at here there are 4 organs that are shown: liver, pancreas, stomach and hypothalamus, and some of the hormones that are starting from, let's say, dopamine starts from the hypothalamus and then it targets the pancreas, right? So there are the source and target tissues that are shown, and then we have obtained these predictions for these hormone gene relations for each of these organs. We can also, there's one model I didn't talk about, you can refer to it in the paper, where we actually predict not just the genes associated with the hormone, but whether it is associated on the source side or the target side. Okay, so that model also allows you to look at the genes predicted on the source side or the target side for each of those hormones and look for enrichments, and largely like for instance insulin, right? So you have this diabetes predictions, and glucagon also you have involvement with diabetes related genes. So you have a large agreement with known genes that are involved in diseases that the hormone is related to. Because there is no other ground truth data set other than the one that we compiled, so we want to look at other ways of validating our signals. So we look at diseases that the hormones are known to be involved in, and then look at those disease genes and overlap it with our predictions. Okay. So there, yeah.
In the last bit, can we also tell about the causality in the direction of interactions?
Yeah, so here the enrichment shown are for all the genes associated with the hormone, but we do have like further level of enrichments where for all the genes we further subclassify it into genes at the source tissue and genes that are involved in the production and genes that are involved in the response to the hormone. But in this particular slide we just show the arrows for the causality, but the enrichments are all for all the genes associated. But we do have predictions for source and target genes. So how much time do I have?
6-7 minutes.
Okay. So because I wanted to talk about the other routes, so we have performed a few other analyses that I would skip and kind of say this is something that the reviewer asked, and then we are quite happy that the reviewer asked. So this is again, these details I'm sharing because obviously as PhD students, as you go through There will be different steps that you go through. You submit a paper, some will get rejected, some will get accepted, and then acceptance is always almost always with a major revision, and so be careful with how you respond. But we are quite happy that one of the reviewers asked, okay, so you have done it on mouse data, but how about kind of experimental validation of these, right? And so in the experimental validation, we are quite happy that they asked, because what we did was we looked at all the, because insulin and estrogen were well studied, there were actually experimental data sets where they take the cells, right? So for instance here MCF7 cells, right? So they take the cells and then in one resting condition they don't expose it to insulin, and in another condition they expose it to insulin, and then they compare the two gene expression data sets to find what are the genes that are changing between these two conditions. So those are the insulin responsive genes. Then we can look at how many genes are there in the reference gene set from these experiments, and we can look at what is our predicted gene sets, and these are robust to different cutoffs that we used, and so different cutoffs we have a significant enrichment between these two sets. So the reference gene set from the experiments and the predicted gene set that we have from our bioembeds method, okay? And these are specifically the method that predicts target genes, so we call it bioembeds TS because these are responsive genes, so we are only interested in genes that are responding at the target site. So we are quite happy about that, and so we have the GitHub resource where you can download the HGV1 data set and then the code and other things necessary to reproduce it, okay? And that's another thing that probably you know by now already, or your PIs would have already told you this. For one submission, we submitted it and forgot to put in a GitHub link, and then two reviewers came back with, where is the GitHub link? We cannot replicate, and they did not trust the study, and then that was rejected. So, so it's important that you work on your GitHub code in parallel and then send the link along so that reviewers have confidence in the results that you're generating. Okay. Okay. So the summary of the first excursion, the second will be obviously briefer than this, but the summary of the first excursion is that a lightweight, because our data set was not too huge, so we have to use just classical ML approaches and a combination of DL based embeddings, right? So these we used BioWordWork, which is not really a deep learning model, but then you can also use BERT and other models for obtaining these embeddings. A combination of these very simple ideas was along with certain discipline in kind of cross validation and balancing was enough to give us a reasonably good accuracy. But one thing that is missing, this is the elephant in the room, is this is big data and biology conference. So where is, literature data is big data, yes, but where is the big omics data, right? This multi-tissue genomic data. Can we use it to further strengthen our predictions, right? So that is something that we also wanted to look at. So, and the main idea there is you first want to construct a co-expression network. So some of you may already know it, but just to make sure that everyone is on the same page, the premise is that if you have intertissue signals between two different genes in different tissues, right? So they may regularly leave a transcriptional footprint. What do I mean by that? If one gene, these are the different individuals where you are measuring this gene Y in tissue B and gene X in tissue A, right? So you have multi-tissue genomic data. So in each individual you have harvested different tissues and you are measuring it. These are post-mortem, of course. Example is the NIH GTEx data. And then if one gene goes up, then the other gene also tracks the gene. If this gene goes down, the other gene tracks the gene, right? So if you look at the correlated scatter plot, they are highly correlated. And this correlation could be due to many reasons. I mean, many factors, confounding factors could drive this correlation. But the idea is that if you have intertissue signal that is visible at this transcriptional level, then we can exploit these intertissue correlations and then do further analysis to figure out if they could give clues about the hormone gene relations. Okay, so that is what we would like to do. We have a kind of another set of work that we do in our lab to try to infer causal relations, try to remove the confounding factors, but that would be for another day. For now, we just look at correlation signals between different tissues, genes in different tissues, okay? And then using this network, using this multi-tissue network which has both this intra-tissue correlations and inter-tissue correlations, right, that you have inferred, can we then figure out important genes involved in hormonal signaling, okay? So that is the question that we have. And this is again joint work with Tarun and RS from Intel that I told, and Sangamitra the postdoc, and Ravi my colleague in the CS department. So we have designed new multi-layer network centrality measures that takes this network as an input, so all of these different edges, It ignores the directionality for the moment. We can include directionality in a future work. It's not too hard, but currently it just takes the network because most of our edges are undirected, and looks at both these blue intertissue relations and these orange, blue intra-tissue relations and these orange intertissue relations, and then we look at how we can use that to figure out key genes involved. Okay. And this is kind of the schematic. So from this multi-tissue genomic data, where across every individual you have profiled multiple tissues, all of the genes, right? So this is a 3D matrix. From that you apply a correlation to get those different tissues. Each tissue is a layer, so that's why we call it as a multi-layer network. Layer and tissue are used interchangeably, right? So there are certain existing methods. If you have a particular network like this, you can have 2 layers, and if you want to figure out genes that are involved in intertissue communication, so direct intertissue communication, you'll probably pick 6 because it has a lot of these dotted interlayer edges. And then, but 2 also seems to be well connected. It's connected to 5 and 6, and then it is connected to the other layer. So we want to be able to exploit both these kind of multi-hop connectivity, right, and these multi-layer connectivity. Okay, so existing methods such as SC will directly look only at these number of dotted edges, the interlayer degree. Okay, so that will not be quite ideal. We want to also pick up node 2 in addition to node 6. So then you can use this multi-hop connectivity, again is studied before, but in the context of single layer networks. So PageRank is an example of it, where you are just saying that the importance of a particular page is a sum of all the pages that is pointing to this page, right? Sum of the importance of all the pages that is pointing into this page. So you have a recursive or iterative equation that you compute, and then finally you get the importance of the different pages based on the importance of the pages pointing to this page, right? So we can use that same concept, but extend it from single layer networks to multi-layer networks. Okay, so that is where the contribution of our work comes in. So we look at if you apply PageRank centrality as is, you will just lose all the layer information, and then you just view it as a giant network, and you apply PageRank centrality. That loses the distinction between the interlayer edges and the interlayer edges and the intralayer edges, right, within and across layer edges. But if you do a careful distinction between these 2 different edges, then we can decompose this matrix. Here there is this layer 1, this is the intra-layer, within layer for tissue 1, within layer for tissue 2. These are the cross layer connections, right? This is from tissue 1 to tissue 2, this is from tissue 2 to tissue 1. These would be symmetric in this case if we use undirected networks. And then you can decompose it into these within layer connections and the across layer connections, right? So this is just an adjacency matrix representation of this graph. And then you can look at extending the PageRank centrality to this kind of a multi-layer network. Okay. And once you have that, then this is kind of the main point I wanted to communicate. I mean, the other things you can refer to in the paper. So the main point is that then you are able to not just get one number, which is 0.50 and 0.45 if you use PageRank centrality on the whole giant network, but you can also understand where that influence is coming from. So 2 influences the other layer, 6 also influences the other layer. They are quite close, but 2 influences mostly due to this local, so that these are in this order. So versatility, local and global. Okay, so the local centrality is high for 2 because it's well connected within the layer, and then it has some connections to the other layer, but 6 is directly connected, so the 0.49, the global centrality is higher than the local centrality, right? So it gives you a way to decompose your overall importance of a node in influencing all the genes in the network in both a local fashion as well as a global fashion, right? Local fashion is useful to understand certain genes that perform their functions within that tissue, and the global ones will tell you which genes are playing key roles in intertissue communication. Okay, and like I said, this is kind of the main point. You can then apply it to, extend it to a setting where you can have a query set of genes. In our case, if you have a hormone, query set of genes will be insulin responsive genes, and you may want to predict insulin producing genes, right? So this may be pancreas, this may be muscle, right? And then you can extend it. So I don't want to go into the details here, but you can extend the formula, and then you can prove certain theoretical properties and apply it to empirical data that is obtained from GTEx. And again we get reasonable results, and I will just skip through this. and just say that these are some of the hormones that we have applied it to. So the insulin, adiponectin, cortisol, and ghrelin, these had enough number of genes both in the source side and the target side in our HGV1 database, so that we could apply it. And these are also the tissues that had enough number of samples in the GTEx data, right? So we need to kind of look at what are all the hormones that satisfy both these criteria. And this is kind of a summary of all the slides that I skipped, which says this is kind of the random 0.5 score, right? So this is area under the recall at cave curve. And then you can see that there are some hormones which have very low number of, so there is now only 2 producing genes. So those are very hard to predict. But if you have at least 10 predicting genes, then you have better predictions, better than a random classifier. With that, I would actually end with my concluding slides. So this is a newly proposed multi-layer or multi-tissue network centrality that extends the standard PageRank centrality. It offers a methodology to find key players in this ICN network that is inferred from these inter-tissue and intra-tissue correlations. And it can also be used to identify genes that are dysregulated in diseases that involve multiple tissues. So we have applied it to multiple brain regions of Alzheimer's disease. And so beyond hormone gene relations, it has broader applicability as well. Okay. And the overall conclusion is these 2 excursions are just just 2 first excursions into intertissue computational biology. There are many more. So if you find this topic interesting, there are many more opportunities and open questions in this area. Okay. Thanks for your attention, and I'm open to take questions.
So first question is, you have used gene ontology for preparing. So gene ontology has also labels, so it's coming from experiment or electronic annotations. So did you do a stratified analysis to check whether you are using a lot of electronic annotations or what, and then kind of curate your data to proceed further?
So in the particular case of insulin and other things, I haven't done a full comprehensive analysis. So that's a good point. And in the particular case of insulin, we have looked at it, and then so many of them are not IEA evidence codes. But for all the hormones, we haven't done a careful check. Yeah, so that is a good check to do. And one thing I can say is that some of the studies that I showed in the experimental validations are recent studies that are beyond the gene ontology annotation downloads that we did. So those things doesn't have this issue. But having said that, yeah, it will be good to look into where those gene annotations are coming from. Yes.
Another question I have is that, so you have used network-based measures for your work. So I recall I've used a database called IHOP, which used PageRank to do the same thing. So did you do a comparison to figure out how it's different?
Yeah, we didn't do IHOP, so we did a lot of other methods that are similar. There's some method called RWRH, so I didn't show those results. So this is random walk with restart. So there is a version of PageRank where you have a set of genes where you can restart from those genes. So if you are trying to predict hormone responsive genes, you set your hormone producing genes as your starting genes, and you keep restarting from there in kind of every random walk that you are doing. So there is a random walk interpretation of PageRank. And so those, compared to those methods, what we have is that those methods do not decompose this versatility into local and global centrality, right? So they are not able to distinguish which genes are well connected because of direct interlayer connections, or which genes are well connected because they're well connected within that layer, and then those genes have other connections, right? So those are not there in existing methods. But if you look at overall area under the curve and other things, then both methods perform similarly, the RWRH and our method. But then in terms of the refined picture it provides, and we show in the case of Alzheimer's disease that those refined pictures give you different pathways that are controlling inter-region dysregulation in Alzheimer's disease through local versus global mechanisms. So it reveals different pathways. So those will be missing in existing methods. Yeah. So those
Thank
are
you.
good points.
Let's now move on to the next speaker is Professor Nagasima. We've not been doing long introductions, so I'm just going to skip over the introduction. So I'm sure you'll have a very interesting